Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy in my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now to our guest.
Hello, this is Richard Jacobs with the Finding Genius podcast, now part of the Finding Genius Foundation. I have Greg Delgoffe. He's an associate professor at the University of Pittsburgh in the Department of Immunology. I'm going to talk about his work there. So, Greg, thank you for coming.
Thank you.
Yeah, if you would, tell me about your research. What are you focused on?
Well, so my research is really kind of focused on the regulation of the immune response. What turns on? I've always been kind of fascinated about why your immune system turns on and off, mainly because we have diseases like autoimmunity where your immune system attacks your healthy tissues. And then we've also have diseases where the immune system doesn't work right, like in cancer, when your immune system doesn't clear those tumor cells away. And so I've kind of always been interested in how your immune system senses its environment. And a lot of people study this. A lot of people study how your immune system gets signals from the various cells of your body. My particular interest in this field is really about food, about how does an immune cell, and I study for the most part T cells, which are kind of lymphocytes that find foreign or abnormal invaders and eliminate them. And so we study how T cells get their food. How do they find their fuel? How do they make their metabolism work? And what we've really uncovered over the course of the last 7 or so years, as I've been kind of running my group, is that T cells are extremely sensitive to the levels of nutrients that are in their environment. And so when T cells get into various tissues, because you remember your immune system needs to go into all different types of tissues and find the invaders, if you will, and remove them. When T cells get into tissues that have different levels of nutrients, it can really change how they function. And so we've been really exploring this from a very basic perspective in some cases, but really the lab has a lot of focus in cancer, about how to turn your immune system on to cancer and how to fuel your immune system to fight cancer cells.
So, these would be some elementary questions, but what are the different cell types that comprise your immune system and where are they made?
That's a great question. So you have, it's kind of interesting when you start talking about immunology. It usually, a lot of times it goes over people's heads because there's just so many different cells and there's not really like an organ. They're kind of everywhere. But, the main players in your body, right, is that you've got this kind of like first line of defense against, pathogens. And you have these cells that exist in your body that kind of eat stuff. We call them macrophages, or we have these neutrophil type cells. And these are cells that basically just kind of eat indiscriminately if it looks weird, essentially. And those are the kind of your first line of defense. We call that your innate immune system, the features of your immune system that are just there, and they look for weird stuff like bacteria or viruses, and they just kind of, gobble. But the real trained soldiers, if you will, in your immune system are your lymphocytes. And so we have two different types of lymphocytes for the most part, maybe three if you're, really getting into it. But the most well-known are your T cells, and they have different flavors and lineages that you can study. And your T cells, they basically kind of run the show. They look for pieces of material that look foreign, and then they kind of tell the other cells in the immune system, those innate immune cells, as well as other lymphocytes, what to do. And so they kind of direct the immune response by secreting chemical messengers that the other cells sense. And they can also directly...
Which cells are these? The ones that orchestrate?
These are T cells. And so these T cells are essentially like the generals of the army, so to speak. And so they can really manage and really direct the entire immune response. And a few of them actually are actually cytotoxic. They actually can go up against an infected cell or a malignant cell and directly kill it. And that's how a lot of your viral clearance and responses to cancer are actually done. And those immune cells arise from cells in your bone marrow, and we teach them how to fight at a school. It's called the thymus, and that's where the T in T cell comes from. Is an organ that you have that sits above your heart, and that's where your T cells are essentially trained to attack foreign invaders. The other one that we can't forget about are your B cells, and your B cells arise in your bone marrow, and they make antibodies. And so those are the cells that you want to stimulate. And your antibodies are your kind of circulating neutralizers. They're basically, they're proteins that find foreign objects, foreign invaders, and kind of bind them and neutralize them. And so those are kind of the players of your immune response. And in cancer therapy, what we've really changed the game on is actually being able to stimulate and deploy these various soldiers in your army, so to speak, to not just fight infection, but to actually utilize your immune system as a cancer therapy. And this has really changed the game in the field of cancer at this point.
Where are these cells created? So you said that T cells, I guess, will be instructed or mature in the thymus, but again, what is responsible for creating these various cells?
So the cells arise. So it's all actually comes from a lineage. So your bone marrow cells are where your immune system comes from. They have all of the progenitors. And as they get different signals during development, as you're, as you grow up and mature, then those cells get different signals. So these progenitor type cells, we call them, they're stem cells. And those stem cells receive they get the right signals at the right time to either form into B cells, to form into T cells, to form into innate cells. And so over the course of your development and maturity, those cells get all of these signals, and it essentially instructs them how to proceed. And that happens throughout life. You're always generating new immune cells, and those immune cells then populate your body in various places. They take up residence in your tissues. They also circulate continuously in the blood, and then they live in certain depots in your body, including your spleen and in your lymph.
Okay, so that's when they go to die. But again, where they're generated is, I guess they have a very complicated life cycle. They're generated in one spot. They move around everywhere. I'm sure there's T cell and T2B communication. There's a maturation for T cells. Then they get chewed up in the spleen and other spots. So it's interesting.
Yeah. So generally what happens is your lymph nodes and your spleen are where they live. They hang out there. And as the rest of your body drains to those sites, that's how B and T cells patrol. They look for things that are in the lymph and that aren't supposed to be there, and thus stimulate the response. And that's all concentrated in your spleen and lymph nodes. We call those your secondary lymphoid organs. That's where these cells live. And then when they get the right kind of stimulation, they see their target, so to speak, then they grow like crazy, and they form a kind of a small clonal army that goes after those pathogens, and they deploy to the blood to go find where it came from. And so that's why your lymph nodes get big when you get sick, right? Because your T cells in your lymph nodes are growing like crazy to try to find that infection and remove it.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit FindingGeniusPodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit FindingGeniusPodcast.com and click Support Us today. Now back to the show.
Are they amassing in the lymph nodes to be deployed? Or why would they be swelling in the lymph nodes, for instance, when you get sick with certain conditions?
Because the lymph nodes that are growing are actually the cells that are proliferating. So when a T cell sees its target, it's gonna proliferate. It's gonna make a copy of itself, and those copies are gonna make copies of themselves, so that you're gonna gain numerically more cells. Remember, those T cells live in the lymph nodes. They're staying there, patrolling essentially the various. They're looking for anything that doesn't belong there, so to speak. And so when those T cells,
so you mean are they monitoring the lymph as a flow through system? Is that what you think they're doing?
That's not what I think they're doing. That's literally what they're doing. Yeah. So as the lymph comes through the lymph node, then that's the place where the immune system gets a crack at things. And if they see something that's not supposed to be there, that cell gets activated and it proliferates like crazy. And as it's proliferating, of course, it becomes, and this proliferation that these T cells do, this division, is rapid. So, we're talking about, a 100-fold expansion of those T cells. So that's going to cause your lymph node to get big. So it's actually full of cells that are getting ready to go out and try to find that infection and remove it.
So they're monitoring the lymph, then they proliferate, then they leave the lymph nodes and, I guess through signaling, go to the spot that needs their attention?
Essentially. And there's a lot of ways that happens. So your lymphocytes patrol your blood. So they enter into the blood from the lymph nodes or the spleen, and they circulate and they look for sites of inflammation and they enter. And so they are able to kind of squeeze in between cells, and they go patrol and try to find those other infected cells as a means to stop the spread of either the infection or the malignancy.
Okay. So I guess they're following, just chemical chemotaxis, like chemical trails, biochemical trails to lead them to the spot.
Exactly. So they generally go to sites of inflammation first. And so that site that's going to be, infected is going to be giving off kind of signals that will look strange, if you will, chemotactic signals indeed. And that causes the T cells, as they're kind of coming by in the bloodstream, they'll attach to the walls of the tissue essentially, and they do what's called extravasation, which is they enter into that tissue from the bloodstream.
Okay. Has anyone been able to, in mice, let's say, induce inflammation in an area that's not inflamed and watch to see if, for instance, T cells would, mice have them, would go to that spot? Can you deliberately, again, call the T cells with signaling and get them to go where you want? Do we understand how the signaling works?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Yeah, absolutely. It's a whole pattern of trafficking. We call it, T cell trafficking. Moves is how T cells get from one place to another, right? So it's not absolutely different types of infections, different types of chemical agents, different types of stimuli can change the pattern of what we call chemokine expression. So chemokines are ways that you get cells to move to one place or another, and T cells become extremely sensitive to chemokines, and that's how they kind of get into tissues, how they know to get into tissues. They also express a number of receptors, so the molecules on their cell surface that actually allow them to attach and get stuck to the sides of blood vessels near inflamed sites. So it's not just like a deliberate movement; it's also getting kind of hooked in to the right places in the body. And those features are kind of how your immune system moves around, and we can take advantage of for various therapeutic applications.
Okay. And in cancer, what's noticed that T cells will they go to a site of a tumor, or then they stop going? Does the cancer totally cut off signaling so that no T cells will go there, or just to reduce the amount?
Yeah, so that's a great question. And so we've actually known since the Civil War that some people have lymphocytes in their tumor. We didn't know what they were at the time. We just knew that it was an immune cell that didn't look like it. But, really, in Civil War times, we knew that if a patient had a lymphocyte in their cancer, they were more likely to do better. They did better than those that didn't. And that's absolutely true to this day. Some cancer patients have T cells in their tumor, and some cancer patients don't. Some cancer patients have T cells in their tumor, but they're stuck on the outside of the tumor rather than infiltrating deep into the tumor. And so we have these immunologic classifications of cancer that are kind of individually known in the cancer field. And we've known for a long time that T cells in your tumor predict better prognosis. So because of that, of those data, right, we've always known that the possibility exists that you might be able to turn on those T cells and stimulate them to attack the tumor cell. And that's been, in the last 20 years has really come to the forefront of cancer treatment. The last, cancer therapy has always been about kind of, cutting it out or poisoning it with chemotherapy. And we're now realizing in many types of cancer, we can use the immune system in one way or the other to attack that tumor, and not just the tumor that you can see, but the distant, maybe invisible metastases that ultimately, that ultimately cause a patient to succumb to their disease. So it's been a really, really exciting time to be an immunologist, because we're now learning that cancer is most certainly an immune-mediated disease, but probably more importantly, it's one that we can really treat with the immune system.
You said that in some tumors, there'll be lymphocytes in them, and that's a better clinical outcome or correlates with it. What about in metastases at the same time? Have scientists observed that, lymphocytes are inside them too? And, what does it look like? Are there more of them in the metastases or less versus the primary?
That's a good question, and it's different from patient to patient. But generally speaking, patients that have a lot of T cells in their tumor tend to have immunologically active tumors, which means even the metastases also have lymphocytes within them. And again, that's all generally considered to be, people do well after surgery, better after surgery than if they have lymphocytes in their tumor than those that do not have lymphocytes in their tumor. But now what we're learning is that patients with lymphocytes in their tumor that would have an immunologically active cancer respond very well to therapies that boost their immune system. And so now we're, again, trying to tackle this problem of, one, how do we, effectively reinvigorate T cells inside tumors? And number two, for the patients that don't have T cells in their tumor, how do we get T cells to their tumor? And those have been some bigger questions. But yeah, T cells see tumors the way they see any other infected tissue. Tumors are most certainly inflammatory, so that can cause T cells to get there. And once they get there, they patrol just like they do any other part of your body.
Well, so the T cells in a tumor, are they active? Are they asleep? Like, do you know what's going on with them? Like, are they eating anything? Like, what are they doing? What are they up to?
Yeah, so generally speaking, by the time a patient has clinically apparent cancer, right? Not something that can't be seen clinically. The classic dogma is that the immune system has tried and failed, right? So the cancer cells mutate and proliferate constantly. And so they're able to develop strategies to either evade the immune system or suppress it. And so generally speaking, when you're looking at T cells inside tumors, these are T cells that have been shut down. And so we have a term for this. We call it exhaustion.
Are they alive? They're just hanging out in there, or what are they doing?
Exactly. They're inert. So these are T cells that are inside your tumor, but they're exhausted. They're being kept alive. They're still alive because they know these T cells can thrive in various different types of environments, but they don't work properly. And
how would you compare their state to T cells in a lymph node where there's no inflammation? They're just hanging out there. Are they the same level of activity, or are they more dormant in a tumor?
No, so the differences in the cells that are in the lymph node, the T cells that are living in your lymph node, they're just waiting. They're waiting to see the target that they're specific. But the cells that are in your tumor, the T cells that are in your tumor, have been actively shut down. They've been actively suppressed. And there are a lot of mechanisms by which T cells are suppressed. But the type of kind of inactivity that happens in cancer is one that's energetic. That when T cells get into tumors, they kind of lose their metabolic sufficiency. And so one of the things, for instance, we uncovered a few years ago was if you take a T cell out of a tumor and you look at it metabolically, you look at its metabolism, it actually has very small and very poorly active mitochondria, which are the organelles that we use to generate energy in our cell. And so as T cells get into tumors, they experience stress that causes their mitochondria to become atrophied, to kind of break down. And this is very causative in their dysfunction. So as they kind of stay in that tumor environment, which has a very, very, kind of harsh landscape, those T cells essentially get the ability to generate energy kind of depleted. And that is really a driving factor in why they don't work.
So, all right. So I guess, scientists have compared T cells inside a tumor versus T cells inside a lymph node. So they're completely inactivated, like you're saying, inside of the tumor.
Yes.
Have people tried various therapies and things to reactivate them in the tumor microenvironment? Like, are there any thoughts on what's needed to activate them again?
Right. So this has been, kind of in the last 20 years, we've really seen a lot of activity in this space. So the first kind of set of really true successes in this field have been really trying to change the signals that those T cells receive. And so this has been called, this is called checkpoint blockade or immune checkpoint blockade, they call it. But it's basically using, drugs that are aimed at kind of blocking and changing the way that T cells see cancer cells. So the thing you have to remember about cancer cells is that they're you, right? Cancer cells are kind of like you gone wrong, but they're still yourself. And cancer cells mutate a lot and they learn, they kind of evolve to turn off your immune system. And the way that T cells are turned off is when they kind of touch up against their targets. The cancer cell turns on these molecules on their cell surface that essentially act as a brake on the T cells and shut it down. And so the immunotherapies of the last 20 years aimed at using drugs that would block those brakes so that the T cell doesn't get that negative signal and it will attack the cancer cell. And so the first one of these was a molecule that blocked one of these brakes on the surface of the T cell called CTLA4. And this drug, it's called ipilimumab, is an antibody that blocks that CTLA4 molecule. And it was tried in melanoma, and you got some really remarkable results that some patients did extremely well on the therapy, such that they were stage 4, fated to die, and they actually experienced complete remissions, complete responses to the drug. And that was extremely exciting. Another drug came about just after, kind of happening in parallel to this other molecule on the cell surface of T cells called PD-1, which also acts as a brake on the immune response. And kind of in parallel, people were developing drugs to the PD-1 brake. And those drugs have had widespread success in a number of different cancer indications: lung cancer, kidney cancer, melanoma, and many, many others now. And you can even combine the two and get an even greater response to these therapies. But the big, and all of these, again, are involved around like kind of telling a T cell that it should turn on, not off, in response to cancer cells. I think the interesting kind of conundrum, though, is that people that go on these immune-based therapies, some of them do extremely well, and they respond beautifully to immunotherapy, and they can have a significant extension of their life. And in many cases, some of these patients are cured. But I think the other side of the problem is that it's really only a subset of patients, really less than 50%, sometimes only 10 or 20%, depending on what kind of cancer it is, will experience that benefit. And the other folks that went on that drug, they don't get any benefit at all. And so that's where, my lab has really been interested in, and many other labs for that matter, is what really defines whether or not a patient's going to respond to these immune-based therapies. And we think that the metabolism is a major part of that the ability of the T cells to get energy and to make their own energy is going to be really, decisive in whether or not that T cell can respond to these drugs.
Well, where do the T cells hang out in the tumor? Are they in the hypoxic or anoxic regions? So I guess they wouldn't survive. Are they on the outer edge? Does that tell you anything? Do they form structures, or are they just kind of loosely hanging out in there?
That's a really great question. T cells are it depends. And I know that that's not a great answer, but when you're talking about cancer and humans, right? Everybody's cancer is unique. Everybody's cancer is different, arise of their own unique constellation of mutations and evolution that have happened in that cancer environment. But generally speaking, when T cells are in a tumor, they are generally, when they come in, some patients have T cells that are everywhere. So we call that a really immunologically active or hot tumor. The T cells are within the tumor and they're ready to kind of attack. And T cells can live in hypoxic regions. It's not good for them, but they can withstand hypoxia and live in the absence of oxygen for a little while. Over time, it can be really deleterious to the T cell, but they can live in some of those areas. But some patients have what are called immune excluded tumors, where the T cells kind of line up around the outside of the tumor bed, but they never really penetrate deep in. And that is because there's a lot of reasons for that, but a major reason is because some tumor cells can secrete a lot of collagen and essentially make a physical barrier to the infiltration of the T cells into the tumor bed. And indeed, there are structures that form, immune structures that can form inside of a tumor. And these are generally considered to be a good prognostic indicator of tumors. We call these tertiary lymphoid structures, or essentially an amalgamation of different types of immune cells working together to form a stronger immune response. So this will be, B cells, T cells, and members of that innate immune system that I alluded to at the beginning, kind of all collaborating to generate a new, more diverse immune response. And so these structures are present in the near and sometimes within solid tumors, and they can actually be very it's a very favorable prognosis when patients have lymphoid structures in their tumor.
Okay, so scientists are seeking to activate them since they're there. Is there any point in culturing T cells if that could be done and, flooding the body with a ton more of them? What would that do to tumors that are hot versus not? Would it make them, would it do anything to them, or it would just not affect them at all, you think?
You're talking about an entire field of immunotherapy. So I was talking a lot, just recently about this immune checkpoint blockade, going after these breaks, right? But kind of in parallel for the last, 20, 30 years or so, people have been exploring exactly what you're talking about, right? Which is, can you numerically enhance a patient's immune response to tumors? And this was really by folks at the NCI in Frederick, the National Cancer Institute, Steve Rosenberg being the main pioneer in this, who back in the late 80s was really taking pieces of a patient's tumor that were removed from surgery and adding a bunch of growth factors and essentially growing the T cells in a dish. And so this is called TIL therapy. TIL stands for tumor infiltrating lymphocytes. And so, these folks, and now TIL therapy is used in a number of different places where you can grow T cells from a biopsy, essentially, of a tumor, a surgical kind of piece of the tumor. You grow them to extreme numbers in a dish, and then you infuse them back into the patient. And there's been some really notable successes of using TIL therapy as an immune-based therapy, but it's still pretty niche. That only some people respond, and so it doesn't work universally, and not all patients have TIL to expand. So sometimes that it just doesn't work. You're not able to harvest enough T cells from the tumor to be able to create an army in vitro. And so,
wait a second, do you have to harvest T cells from the tumor, or can you harvest them from lymph nodes where they haven't gone to the tumor yet and been, put to sleep?
That's a great question. And it kind of gets back to immunology. And that's that T cells are individually, they're clonal. So that means that every T cell expresses a receptor that finds a particular kind of target. And so the T cells in your lymph node, they are going to be specific for all kinds of things, not just tumor. And so by isolating the T cells from the tumor, you're getting the T cells that are most likely specific for tumor, if that makes if that makes sense.
But is there any problem because they've been silenced? Are they affected permanently, or they're still better because they've encountered the tumor microenvironment? They've kind of gone maybe 1 or 2 rounds with the tumor, but now they've been silenced. So are they better than ones that have never seen the tumor directly or not? Is there a trade-off there? Is what I'm asking.
Absolutely. Yeah, absolutely. So you bring up a very, very good point. And that is, some patients may have T cells that are just completely exhausted, completely inert. And so they're not going to be the ones that are going to be able to grow TIL very well. Or if they are able to grow from that biopsy very well, they won't be effective. And other patients may have T cells that are less exhausted, and they may do better after you expand them in a dish. So yeah, you're absolutely right. It's tough to tell, but there's no way to reliably get those kind of tumor reactive T cells from the blood or from the lymph nodes. So you kind of have to start from the tumor. But it's an interesting thing that you bring up, because some people have that exact same kind of conundrum, right? Which is, why are we trying to get these T cells from the tumor where we know they've been rendered inert? Is there a way for us to make T cells see tumor on their own, right? To take healthy T cells and use those T cells and kind of redirect them towards tumor target. And that was really the advent of what are called chimeric antigen receptor T cells, or CAR T cells. And these are T cells that are taken from the blood. So these are T cells that are healthy. They haven't been rendered inert inside that tumor environment. But now they're engineered with a receptor, a protein that's going to sit on their cell surface that recognizes an antigen that's going to be present on a tumor. And you use genetic engineering to put this receptor into T cells, thus arming them and directing them towards a tumor. So you do that kind of manipulation in vitro, in a dish, and then you grow up the T cells and you deliver them back to the patient. And these types of T cells, CAR T cells, have had extreme success in certain types of cancer, namely liquid cancers or hematologic malignancies, most notably in B-cell leukemias. And the T cells in these environments, they can do quite a lot when they're given this, they've been given specificity for a tumor. And it's been really exciting to watch that field kind of progress. And I think it was in 2017, this was the first FDA approved gene therapy, essentially, where a patient with B-cell leukemia could send out their blood. It would get transformed into tumor fighting T cells, and those T cells would be delivered back, and that patient could experience very, very durable and remarkable responses to that therapy. It's been really exciting, but we haven't been able to make CAR T cells yet against solid tumors. They work really well in certain types of hematologic malignancies, but we haven't been able to really outfit them.
Not you, but are scientists taking T cells that are within a tumor and culturing those and then re-injecting them back into the tumor, whether CAR T or some other modification? Or are they taking ones against that are hanging out in the lymph node that are naive, and then CAR T, putting this weapon on them, this CAR T, or, whatever I call overlay over them, and then sending them into the tumor? And are they injecting them into it, or are they just letting the circulatory system bring them to the tumor? How does the therapy actually look?
Yeah, that's great. So TIL therapy just grows them from the tumor, as I was mentioning before. You just add growth factors and the T cells will expand in vitro. But these CAR T cells are made from healthy T cells. These CAR T cells are made from the blood, which is mostly, healthy circulating T cells that can be easily accessed by a doctor, right? So they can harvest the T cells from a blood sample, and they are that the CAR is essentially engineered into the T cell and then grown up. So then they grow those T cells in vitro, so you have a lot of them, and then they're injected back into the patient. Now, here's the big thing is these T cells are delivered intravenously, because you don't just want them to go to a tumor. You want them to patrol your body just like any other T cell would do and find all of the tumor, not just the one that is obvious, but the distant, metastases or even micrometastases that you can't detect. And that's going to really, that's what's really important, is that, these T cells that we're using for therapy, these T cells essentially become like a living drug. right? Those T cells are going to always patrol your body for their target. And that's what we want. That's why we get vaccinated. That's why, you only get the chickenpox once, right? Because your T cells then patrol your body for that virus if it were ever. And that's kind of what we want in cancer, right? We don't want to worry about cancer coming back. So if you can educate your immune system one way or the other, maybe that's the T cell therapy, maybe that's checkpoint blockade and reinvigorating that immune response. What the goal is to educate your immune system such that whenever that tumor were to come back, those T cells will attack.
I don't know, has anyone ever thought to try to inject it, again, directly into a tumor?
Yes, actually, there's an entire class of intratumoral therapies that are used. Now, you can inject all kinds of stuff into tumors. And that's great for patients that have accessible tumors. That's why a lot of the immune-based therapies were originally done in melanoma, because number one, it's very immunologically active, and number two, the metastases the cutaneous metastases are easily accessible. But some patients don't have accessible tumors; they have to be ultrasound guided, etc. But yeah, a lot of therapies are delivered intratumorally, things that are aimed at stimulating the immune response. Many different types of therapies are used. Chemical messengers for the immune system, things that are designed to destroy tumor cells, including things like modified viruses that are that can replicate in tumor cells and cause them to lyse.
But for CAR T, where does the choice of the CAR come from? And does it affect preferentially all the tumors, or are they different enough where it could affect only the primary or only metastases?
So the CAR, right? The CAR, so CAR stands for chimeric antigen receptor. And to basically just break it down, it's an antibody. So it's a piece of an antibody that would normally be floating around in your body that's designed to neutralize something. And you take a piece of an antibody that recognizes a tumor and you make a chimera out of it. So you make a receptor that on the outside looks like an antibody, but on the inside has all of the parts that would stimulate a T cell. So it is going to act. So it acts like a T cell's kind of native receptor, how it would normally see a pathogen, but instead that antibody fragment on the outside can be triggered by a tumor.
I ask because I've heard, 80 million times that tumors are heterogeneous and the metastases are even more heterogeneous than the initial, primary. So how do you know what to target? How do you choose? Are there many targets? And again, does it apply to all cancer cells within someone?
Yeah, you bring up a very good point. And that's the main reason why I think we haven't been able to deploy CAR T cells against solid tumors, is because there isn't a good target. There isn't a good target that is not only, it can't just be on all tumor, which is what you want, but it also needs to not be on anything normal, right? You don't want that thing on the cell surface to be on anything that's on your normal tissues, because then the T cells will attack those tissues as well. So the current CAR T cells that are used actually target a molecule called CD19, and a CD19 is on the surface of B cells. So, and it's used to attack B cell leukemias, a cancer of your B cells, right? And so the reason why CAR T cells have been so successful is because they used an antibody that recognizes B cells. And it works extremely well at depleting B cells. But it also means that patients that get CAR T cells also don't have any healthy B cells. They don't have B cell cancer, but they also don't have B cells either. Luckily, you can live without B cells. You don't need B cells to live a normal life. And many patients that have received, these remarkable responses to CAR T cells in B cell leukemias will live their lives without any B cells. Those T cells, the CAR T cells inside them will always keep hitting the B cells. So that's why we've had success in heme malignancies, is because cancers of the blood have very defined markers that you can generate these CARs to. We do not have that luxury in solid tumors. There hasn't been yet an antigen that's present on solid tumors that either one is going to be on the tumors that you want, and number two isn't on a normal cell that you can't afford to lose.
So I guess then liquid tumors are a bit less heterogeneous than solid tumors, And also the structure too, I guess, would act as multiple layer barriers to get to the, to the heart of the tumor as well.
Absolutely. And certain kinds of hematologic malignancies that have more of a niche, that have more of a reservoir, maybe they live more in the bone marrow, they've been harder to target. And other hemalignancies that have a lot of heterogeneity haven't been targeted yet well with. So it's not a one size fits all kind of thing, but right now people with certain kinds of B-cell leukemia and certain kinds of B-cell lymphoma, they can go on, this CAR T-cell therapy and really expect to see some major benefits. But it's not the case for the rest of the cancer world.
Well,
that's why we have these
yes. Do T and B cells release, I would guess they release extracellular vesicles. Is anyone studying the signaling that goes on amidst the T's and B's and T's that are trained in the thymus and, T's when they get in the vicinity of a tumor or inflammation? I would think there's a whole bunch of communication going on, maybe even quorum sensing amongst T cells. Are there enough of us to do a certain job, that kind of thing?
Absolutely. So T cells are, by their nature, are signal integrators. That's what they do. They receive signals from the cell surface. That's, when they kiss up against the target, or when they receive chemical messengers, which we call cytokines, right, that bind the receptors and deliver signals to the inside of the cell. So they're signal integrators. They take all that stuff, they make a decision, and then they start secreting their own cytokines to the other cells in the milieu. So T cells absolutely collaborate with one another. They work with other cells, and they use either cell surface communication or they use soluble mediators to deliver those signals. So immune cells do utilize like exosomes or extracellular vesicles to deliver signals as well. They're a little bit less studied than these other kind of chemical messengers, but these things absolutely do occur. So when T cells get near a tumor, they absolutely, play this role of talking to one another, proliferating when they can, and then getting inside that tumor environment. But that's when things go crazy, because no matter what kind of signals they're getting, right, whether or not it's accelerators in the form of, stimulatory cytokines or, the right kind of positive signals to kill a target cell, or whether or not it's brakes, these co-inhibitory molecules like CTLA4 and PD-1, like I was talking about earlier, that kind of turn off the immune response. There's no fuel in that environment. There are no metabolites that are present because the tumor has kind of eaten them all. And so when those T cells get into that tumor, regardless of the signals that they're getting, there's no energy, there's no fuel in that environment. And so the T cells can't really get going. And I think, that's something that, again, in my lab, we study a lot of, trying to understand what are the metabolic requirements for an effective T cell response against cancer, and how can we deliver those metabolites or those metabolic signals to T cells in a way that they can thrive in that environment.
I mean, you said at the very beginning, depending on the nutrient composition, the T cells act very differently. So again, in a tumor, you got hypoxic, anoxic, and fully oxygenated regions. Where do the T cells tend to become trapped or inactive or hang out? And, what kind of nutrient mix can you get once you're within the confines of, let's say, a solid tumor? Maybe the outermost layer, I don't know. I mean, I don't even know how the tumor gets nutrition. So again, what kind of dynamics go on there?
Yeah, there's a lot of very complex dynamics within a tumor. And that is because, tumors evolve over time, right? So they develop their own blood supply, right? So they're angiogenic, but that blood supply is not correct. In other words, it's not the typical blood supply that you get. It's actually this kind of tortuous vasculature. And so the tumor is really, really, really well perfused and super oxygenated in some areas of the tumor. And then other areas of the tumor are very, very hypoxic. And this makes a big difference for kind of what the whole metabolic landscape is inside of the tumor microenvironment, as we call it. And so a T cell, depending on whether or not it's just entered in from the bloodstream, things might be fine, right? Perfectly perfused, plenty of nutrients, but that's not where the real problem is. The cells might be, the tumor originating cells, maybe, within these hypoxic regions. And so, like I said before, T cells can withstand being in low oxygen tension, but it's the other features. It's not just the low oxygen. Tumors, of course, eat a lot of sugar, right? They're dominant metabolizers of glucose. That's what we use to find tumors. In fact, if you get a PET scan, right, what a PET scan actually shows you is the glucose uptake of cells in your body. And so we know that tumors are extremely metabolically active, and so they take up a lot of glucose. So that's how you find tumors in a full body scan. Now, that has major consequences because T cells also need glucose to function, just like all of our other cells in our body. And so it's a land grab, so to speak, for certain types of metabolites, not just glucose, but amino acids, lipids in some cases. These are all in competition. But I think what complicates matters even more is that it's not just, and this is something we've really discovered a lot of in the lab and many other groups as well, is that it's not just that the tumor kind of eats all the good stuff. It's that it's also in certain areas of the tumor, remember that vasculature is not great. So the tumor actually spews out all of the toxic byproducts of that metabolism, all the waste products. And so it's not just that the tumor eats all the glucose. It's also that it spits out all this lactic acid. And that lactic acid can really dampen the immune response. So like, it's not just lactate, of course, it's many, many other catabolites of all this heightened metabolism that's happening inside the tumor. These can act as very, very potent immunosuppressive agents. So I think a lot of things now going forward are actually aimed at kind of tempering and removing those kind of toxic metabolites from the tumor and making it a little less harsh on the immune system as well.
I would guess there's people that study the outermost cells or the layering of the structure of tumors and what can transit through them and what can't.
Yeah, I think there's a lot of folks out there that really try to, figure out some of the, geography or topography, if you will, of tumors. It's different. Like I said, everybody's cancer is different. You can study it in mice, of course, in models of mice and in preclinical models, which have functioning immune systems, and a lot of our immunotherapies were originally designed, and tested in animals, in mice. But the way that mice get cancer is usually experimentally. You either put tumor cells into mice and they grow, or you have to induce cancer rapidly so that you can study it in a reasonable amount of time. And so they don't have the same features that your typical your typical human tumor would look like. But that being said, there's a lot of interest in understanding how different parts of a single solid tumor or other individual tumors would, how immune active some areas are versus others. And I think the study of that, of understanding, where there's a lot of immune activity versus where there isn't, these are actually really helping scientists understand the nature of immune regulation in cancer. But I think probably more importantly, it's starting now with the aid of kind of computer-assisted pathology. It's actually helping doctors make decisions about immunotherapy. Is that by kind of getting a good look at a patient's tumor before you start treatment, you can actually try to design the right kind of immunotherapeutic approach to take advantage of that patient's own tumor, so to speak. Does this patient have an immune active tumor? Do they have an immune excluded tumor? Do they have any immune infiltrate at all? Where is that infiltrate? And by gaining some of that information, you can actually help guide the clinical decisions for therapy.
Is there any way to tell the level of T cell infiltration without biopsying the tumor? Looking for just blood markers, or looking in, some of the, let's say, adjacent lymph nodes and at the condition and number of T cells there?
Yeah, it's very difficult. It's very difficult so right now we only have a couple of, very bespoke, if you will, ways to tell whether or not a T cell can see a tumor. Meaning if you just by looking at it, you can't really tell what it responds to. You have to kind of look indirectly to figure that out. And so, that's a big hurdle to overcome. It doesn't mean that we can't do it, but right now it's very difficult to get kind of like the non-invasive way of doing it. But yeah, so right now, if you really wanted to figure out if a person's got an immune active tumor, you have to look in the tumor and look at a tumor biopsy to be able to figure that out. I don't think that's going to be the case forever. And I think our advances in being able to predict how T cells behave and what we can do with, based on sequencing and understanding the genetics of an individual T cell, I think we will be able to get to the point where you'll be able to tell whether or not a patient has an ongoing immune response to their tumor, basically based on a blood sample.
Have you guys worked or seen anyone work with the tumor organoids? And can the tumor organoids house T cells and silence them?
Yes. I think, the organoid field is really great because it gives, you always have to play the problem is whenever you're trying to do conclusive science, you're kind of stuck in this field in cancer, right? You're working with tumor biopsies and you're just trying to see what you can see, or you're working in mouse models where, there's going to be obvious issues translating that to humans, because you have to use their speed and, it's not proper cancer the way that it naturally develops. So, somewhere in the middle, what can you do in vitro? And you can't just, culture T cells with tumor cells. You don't really get the same kind of immune suppression the way that you would in vivo, in an animal or in a person. But organoids are really interesting because those tumors do start to form a little bit of an environment. And some of the time you can actually seed those organoids, those little tumor spheres or whatever you want to call them, with T cells. And indeed, absolutely, T cells that are specific for that tumor, they will infiltrate into the tumor and become inactivated. That's part of the, there's a lot of labs that do that kind of analysis, taking patient T cells, taking patient tumor biopsies, and actually trying to replicate a lot of this in the dish using these kind of three-dimensional culture systems.
Can you fluorescently tag or radioactively tag T cells? And then in vivo, like in a mouse model, for instance, cultured T cells, mark them, release them, and see how they interact and how they enter into a cell how they enter into the tumor and how they become changed. Is there any way to do this again in a mouse model?
Yeah, absolutely. This is what we do all the time in our lab. This is called adoptive transfer. Mice are great because you can track their T cells you can move T cells from one mouse to another. And if they're on a different enough background, you can follow those T cells using a number of different technologies. And so, yeah, we do this kind of stuff all the time. We can take T cells from one mouse and modify them or not. And we know that they're going to be specific for tumor antigens because we've engineered them that way. And then we can infuse them into a tumor-bearing mouse and study their behavior as they get into the system, go to the lymph node, ultimately migrate into the tumor, and then become inactivated. And by using that trajectory of those T cells as we move them, we can see kind of what are the features that drive the immune suppression or immune inactivation in that environment, and then go back and make genetic changes to the T cells and try to prevent that from happening, or different things that you can add to the mix, different treatments of the mice, different genes that you can express or delete in the T cells, and ultimately see kind of what is were you able to avoid that immune activation or exhaustion, or did you uncover some of the pieces that are a part of it? And so that kind of work is something that we, is part and parcel of a lot of cancer immunology labs, using T cells from one mouse and transferring them and being able to track them over time and track their activity.
So T cells can burrow into a tumor no matter its structure, or there's some tumors that keep them out completely?
There are different types of tumors that exclude immune cells, and that can happen through a couple of different mechanisms. One I mentioned earlier is they can actually generate physical barrier. They can generate, some tumors are derived from cells that can make collagen, or they can make, a really kind of dense structure so that it actually prevents T cells from burrowing through. Other types of tumors aren't. They just aren't. They don't have that programming, so they don't generate that kind of physical barrier. Other types of tumors can create metabolic barriers, like we talked about. They can generate extreme hypoxic regions where T cells will get in, but then they stop moving. They run out of energy, so they kind of get stalled at the outside. I think that's kind of the take-home message when you're talking about cancer immunology, is that the tumors have the benefit of time. They're persistent. When people have cancer, they've had that initiating event that caused that initial neoplastic event happened a very, very long time ago. And it's been this process of trial and error that cancer cell has used to develop all of these different suites of characteristics that allow them to evade immunity. And, some cancers go rogue. They stop presenting antigens, so they can't be detected by the immune cells. Some create more like immunosuppressive structures, like these physical barriers. And some cancers, kind of harbor T cells, but kind of deplete them over time, like a war of attrition. And so we have to study all of these features of cancer, identify the kind of immunologic problems, and that's how we can design effective therapy, by really kind of understanding all of the potential mechanisms of cancer immune evasion and suppression, and realizing that these are not common. They're not like, it's not that all cancers do all those three things. It's that everybody's cancer arises differently, and we need to learn better what's happening, and we need to also know how to find those features in patients. So again, that we can guide the right decisions to effectively, boost these, find a way to either numerically augment in the case of CAR T cells or that TIL therapy, to reinvigorate in the case of, checkpoint blockade, or even, even more heroic measures to kind of, reinvigorate a patient's immune response to their cancer.
I interviewed a researcher named Florencia McAllister, who's studying pancreatic tumors, and she saw that the pancreatic tumors had a different localized microbiome than the rest of the pancreas. For liquid tumors or for T cells themselves, or for solid tumors, I would think they would have their own, again, localized microbiome. But has anyone seen a microbiome of the blood, or even individual T cells themselves? Do they have any hangers on, any microbes that hang out with them and act as kind of maybe like a quick version of the microbiome for them?
Yeah, so that's what you're talking about is a kind of a fascinating and really hot area of research right now. And I know that work very well, actually. But yeah, it's been, understanding how your microbiome and your immune system interact, because that's a very, very active environment. A lot of your immune system is in your gut, and keeping that, the nature of that place at bay, and also being able to derive benefit from those commensals that live in your gut and help you digest food. But yeah, it's actually a few years ago, it was found in that kind of checkpoint blockade immunotherapy that patients that have a certain kind of microbiome tend to do better on therapies than patients that have a different kind of microbiome. And that's been just in their guts. And that actually the gut microbiome could help predict whether or not patients would respond to immune-based therapies. And since then, it's been, really explored about how the microbiome does interact with immune and specifically the anti-cancer immune system. And, tumors can have their own microbiome, especially if they're at barrier surfaces. So they can have, bugs that live within them, and you might be able to conscript, for instance, tumor-specific immunity to those places. We know that, there's also kind of bystander effects, that an active immune response against a bug could help promote a nearby immune response against a tumor. And then finally, the reason why we have microbes is because they help us digest our food and provide metabolites that we cannot make ourselves. And so again, I'm a metabolism person. I can't really not think that a major component of how microbes interact with our anti-cancer immune response is by creating fuel that we can't make, and that T cells may like. And so that's a feature, I think, of this interaction that's really exciting.
Yeah, like when you mentioned tumors that spit out a lot of lactic acid and undergo a lot of that fermentation process, I would think in short order they would attract microbes that like to feed on that, or some of the other metabolites generated. Or again, a tumor in general, if there's a hypoxic or anoxic region, I was sure it would attract microbes that wouldn't normally hang out because they can deal with the anoxia or the hypoxia.
Absolutely. And so that depends critically on, whether or not those bugs translocate from the gut and ultimately can make it, get into the bloodstream and then ultimately find that tumor, or whether or not, what the pre-existing kind of landscape was, right? And again, if that tumor is at a barrier site, like in the skin or in the mucosa, then it's going to be more likely that there's going to be populations of that tumor with bugs. The other possibility, of course, again, is that kind of translocation into the gut. And that, of course, can happen for a number of different reasons. So yeah, so I think this interaction between microbes, their metabolism, cancer cells, and then the ongoing immune response to all of these features, I think is going to really help us define kind of like a unified model of how we think not only tumor-specific T cells, but also adjacent T cells that are responding to maybe a different kind of, foreign body, like a commensal microbiome or even a pathogen, how those things are all going to interact with one another. And I think it's really an exciting future to see how those things all work together.
Yeah, excellent. Well, Greg, what's the best way for people to find out more about your work? Where can they go?
So we obviously have a website, so you can find it at delgofflab.com. You can also follow me on Twitter at delgofflab, where we're very active talking about all the work that we're doing, exciting papers that we're publishing, and all the fun stuff that we get to do while we're trying to trying to decode what's happening with our cancer immune response. And always, you can always just reach out. I'm always happy to chat about these things.
Well, very good. Greg, thank you for coming on the podcast. I appreciate it. It's been very interesting.
Great. Thank you, Richard. Talk to you soon.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.